Hello AP Biology students, we're going to go over the third section in Unit 5. And in this section, we're going to talk about Punnett squares. Before I get into Punnett squares, I want to remind students that Gregor Mendel from our last section didn't make Punnett squares. It was a British scientist later on, years later, that was named Reginald Punnett. He actually wrote a book called Mendelism, but he was basically looking at how traits are being passed from parent to offspring as well. This is a Punnett square, and it's a tool we use for predicting the traits of an offspring. The boxes here in the middle represent possible offspring. The alleles here on the side and on the top represent the possible alleles that the parent could give. Again, here's another way of looking at it. You can see here the letters on the side are one parent's possible alleles they could give. So this mother's genotype is big T, little t. Now remember, a mother is only going to give half of her DNA to her offspring. So in this case, this mother can either give the big T or the little t. Now the father has the same genotype. You can see it's big T, little t. However, again, you're only going to get half of your genes or half of your DNA from the father. So the father is only going to be able to give the big T or the little t to the offspring. Again, here's another way of looking at it. You can see that the alleles represent the different genes that a possible sperm or egg may have. The boxes here in the middle represent possible zygotes that could be formed. It's not saying that each one of these boxes or each one of these zygotes will be formed. It's just saying that there is a possibility of this zygote being formed. Now, in a monohybrid cross, there is a cross between a single trait. For instance, like our last section, we talked about tall pea plants versus short pea plants. We're not looking at any other traits other than this just one trait, tall versus short. If you remember in the first cross that Mendel did, he crossed a true breeding tall pea plant with a true breeding short pea plant. What he was actually using was something called a homozygous dominant pea plant and a homozygous recessive pea plant. You can see when these two are crossed, they're each going to only give one allele to their offspring. So in this case, the big B plant can either just give one big B or this big B. And the short pea plant, little b, is only going to be able to give a little b. So 100% of the time, the offspring are going to be big B, little b. You can see here, there's no chance of being little b, or big B in the offspring. Which means, since the law of dominance states that as long as you get one dominant allele, you're going to get the dominant phenotype, all of the offspring are tall. But again, all of the offspring are heterozygous. When Mendel crossed two of the heterozygous genotypes or the F1 genotype, again, we have that big B, little b on the top and big B, little b on the side. The offspring in the F2 generation could either be big B, big B, little b, or little b. But like the Punnett square shows, 1/4 of the offspring should be big B, or 25%. 50% of the offspring should be big B, or 2/4. And lastly, little b is only going to be seen 1/4 of the time, or 25%. If we look at the phenotypes, again, big B and big B are both going to be tall. The only chance that the offspring will be short is this little b genotype. This means that 3/4 of the offspring are going to be tall, while 1/4 are going to be short, or that 3-to-1 ratio that Mendel saw in the F2 generation. So again, just as a quick recap, Mendel crossed a tall and short pea plant in the P generation that were both homozygous. All of the offspring in the F1 generation were heterozygous but tall. In the F2 generation, there was a 3-to-1 ratio of tall to short. But remember, 1/3 of the tall pea plants were homozygous dominant and 2/3 were heterozygous. All right, this is a bad one, but what did Gregor Mendel say when he founded genetics? Whoopee, because pea plants, that's a bad one.